So I think we're going to get started on time here, so I'm going to do my introduction. So it's our pleasure to introduce Dr. Natalie Surkova today. Natalie very graciously stepped in at the last minute to fill a vacant spot, and she should be commended for that. And so Natalie has a very worldly education. She received her bachelor's, master's, and PhD at universities in France, the Ukraine, and in Germany, where she studied biochemistry and physics. And then she came to do her postdoctoral work at the University of California, San Francisco, where she continued her work on biochemistry and physics, and it's there, and pharmacokinetics, where she got interested in animal imaging. And we were very fortunate to recruit her here in 2002 as an assistant professor, and she's risen through the ranks and became a tenured professor in anesthesiology in 2016. And at present, she has appointments in primary in the Department of Radiology and also secondary in anesthesiology and radiation oncology. But most importantly, Natalie is the director of the Cancer Center Metabolomics Core, the Cancer Center Animal Imaging Shared Resource, and also for the CCTSI Institute Imaging Core. And many of us know her in that regard, and we are very fortunate to have really high-tech imaging equipment at this university and be able to do the things that we do with animals. And Natalie has over 120 peer-reviewed publications. She's extremely collaborative. She has co-published with dozens and dozens of researchers on this campus. And as I mentioned, she has kept up amazing instrumentation and is always pushing the limits to do new tracers, new metabolomics. And I won't say much more about that or I'll get myself in trouble. I'll let her talk about that. And she is the PI and co-PI on several NIH grants. And most recently, she wrote a very large grant for a new Bruker 9.4 Tesla MRI scanner, which I hope we hear about today. And that was quite an achievement. And I hear that we may be in the works for a new IVUS imaging equipment as well. And she has beautiful pictures. She's always fun to work with. And I'm excited to hear what she's working on. So I'll be quiet and let her give her seminar.
Well, good early afternoon, and indeed, thanks so much for introduction, Dr. Sartorius. I'm going to talk about our new baby. It's our 9.4 Tesla MRI, so it's ultra-high field MRI, which is wonderful for the mouse imaging, especially. I was very excited because using the old system, the brain images were not really of the supreme quality. So it has changed after the introduction of the 9.4 Tesla MRI, which has happened in September 2018. So because was just such a huge difference in the brain imaging, we actually, I would say probably 60% of our collaborative work, which we have done so far, was with the neuro-oncology team, because the pediatric neuro-oncology team, because they were very desperately waiting for this system. And in less than 4 months, we probably scanned, I would say, hundreds of mice now, right, Jenna? And we already actually submitted 3 papers, and one already has been published at the JCI. So it's really like blitzkrieg. It's going so fast and so great. So today I'm going to talk about the advanced MRI to characterize tumor habitat and microenvironment in orthotopic mouse model for pediatric brain tumors. And my first disclosure is that it's really not a one-person work. It requires a dedicated team of the MR imaging expertise, of the animal imaging expertise, which my group is providing, and also obviously for the mouse neuro oncology and PDX model expertise, which is provided by Dr. Rajiv Vibhakar team, Dr. Adam Green, and Dr. Nick Foreman. And unfortunately, all 3 of them are out of town today. So I hope that some team's members are here. But it was really last minute when I decided to give this talk. So the background is, and I don't know if you know this, but the brain tumors, even though it's obviously one of the deadliest human malignancies, is actually not the most common brain malignant disorder. So 90% of all brain lesions are metastases, and this is why I'm working very close with Carol Sartorius, Dr. Sartorius. We're also imaging the brain metastases, but we're going to talk today about 10% of all bad guys, and particularly it's the brain tumors. And in adult population, the brain tumors make 10% of the brain lesions. And even though there's such a low number, but they are definitely devastating with regards to their morbidity and mortality rates. In children, however, brain tumors, because there's not much of metastatic disease in children from the other solid tumor sites, brain tumors present 1/4 of all cancer deaths, and they also are second most common malignancy in childhood after leukemia. So this is leukemia, and those are brain tumors. And more information about the survival rates and so on, you can get from the Central Brain Tumor Registry of the United States. It's called CB Truth. Here's the website. But in overall, what they report, that there is a significant increase in brain tumor incidence over 30 years, especially in children, and probably it's caused by several changes in etiological and also genetic factors. Medulloblastoma per se, especially in the very young children, remains the most common pediatric brain tumors. It's approximately 18 to 20%. High-grade gliomas, which is usually the most common in adult, present the second most common brain tumors in children. And finally, ependymomas, which is the third often occurred brain tumors in children, is 12%. However, they're one of the most deadliest and very, very bad clinical outcome for ependymomas. So the MRI per se is the gold standard for the diagnostic management and therapeutic management of the therapeutic follow-up of the treatment response in the adult patients, and we also use it all in the pediatric patients. And there's a different opportunities which MRI can provide for brain tumor patients. We can do anatomical imaging, physiological imaging, even functional imaging, mostly to plan radiation treatment and surgery treatment. And those modalities, those protocols, MRI protocols, allow you to see inside of the brain and without taking a biopsy. So anatomical imaging usually has high resolution, which in humans is approximately 1 millimeter, and you can see and also measure the dimensions of the tumor. For example, this is glioblastoma in an adult patient, so you can report the tumor diameter square, and you can even calculate the tumor volume. In addition to this, MRI can also give you an idea about the physiological microenvironment and properties of the tumor. For example, by MRI, using mostly diffusion-weighted and perfusion-weighted imaging, you can see the central blood flow. So the anatomical lesion is right here, and you can see that there increased blood flow in the same region, meaning the angiogenesis in the tumor. And you can even calculate blood vessel density. Finally, functional MRI is used very often for the surgery planning, because if you want to resect a tumor, you have to make sure that there's no important centers for example, any kind of cognitive functions. This is, for example, how we do sentence generation. So we ask the patient to say something meaningful, and we see which regions in the brain are activated. So for this patient, fortunately, there's no overlap between the sentence generation centers and the tumor. We can also do different nerve tracking and just make sure that the resection doesn't come close to those tracking. So this is all good and great, and it's very well established in the adult patient population. But there are some challenges with the children. First of all, functional MRI is very difficult in children because we have to sedate them, especially small children. And obviously, anesthesia sedation will compromise functional MRI in response to the stimuli. And also, we always try to do, and sometimes those MRI on adults take over an hour on the imaging time. It's almost impossible to do it in pediatric patients because most of them are sedated. We don't want to keep them a long time under anesthesia and in MRI scanner, which is quite intimidating per se. We also need high resolution in adult brain, preferably no gadolinium contrast, because in the past couple years, there are some significant safety issues with the gadolinium contrast, so we do not want to inject them into a child, and most actually protocols are gadolinium based. So we also need to try to characterize tumor subtypes because they are absolutely different from the major adult brain malignancies. And we need to characterize tumor microenvironment, and especially for the patients who undergo radiation treatment, and especially with the increasing application of immunotherapy and checkpoint inhibitors, we need to have an imaging protocol which can see the radiation treatment response and inflammation. And because of all of this, the goal of our study with our neuro-oncology team was to develop, and we call it MAFI, almost like mafia, multi-parametric advanced fast imaging on mouse PDX models on the brain. And we will do it so without using of the gadolinium contrast, which for me as a MR physicist represents some challenging. So we also wanted to classify and characterize different pediatric brain tumor types, 3 major pediatric brain tumor types based on our appearances of the MRI scans. And finally, we also establish a MAFI classifier based on the angiogenesis, peritumoral edema, and metastatic spread for each pediatric brain tumor type. So what the mouse model has been developed in our collaborator laboratories, and actually most of them was performed by Dr. Angie Pierce. So they're using female severely immunodeficient SCID mice, and they do intracranial orthotopic inoculation of pediatrics patient-derived xenografts. So those are child-specific tumors removed during surgeries. And we had 3 different tumor types. We used medulloblastoma, which was inoculated into the 4th ventricle. So far we analyzed and processed 11 tumor type, 11 medulloblastomas. And next tumor type was a so-called DIPG, is diffuse intrinsic pontine glioma, which was injected into the pons. It's where it's supposed to be. 8 mice were analyzed, and ependymomas were injected also in the 4th ventricles and go straight into the cerebellum. It's actually the majority of the animals were with ependymomas. The tumor size which we were able to detect was as small as 0.4 cubic millimeters, so it's pretty impressive. It's almost like a dot somewhere in the cerebellum of the mouse. And the biggest tumor size which we just analyzed were 78 cubic millimeter in the brain. And we always did isoflurane anesthesia during MRI session, but no contrast. And this is a little mouse in the induction box, and then you put it into the MRI holder, put the mouse brain coil, and the mouse is still anesthetized with the gas mask, and the mouse goes into the MRI scanner just like a patient. So for the MRI, we use ultra-high field scanner, which is a 9.4 Tesla with the mouse head array coil. And we use non-gadolinium MAFI brain protocol, which consisted of the high-resolution T2 scans. It's three-dimensional coverage of the brain, and we needed only 5 minutes to do it with this high field, and the spatial resolution was about 50 microns. We also used so-called inversion recovery flare MRI, 2 minutes, to precisely quantify edema in the brain, and we used diffusion-weighted MRI, which was only a little bit more than 1 minute, to once again to confirm the edema, necrosis, and also blood vessel density. So total of 8 minutes scan, which is a very short time, was applied to assess all those parameters, which are conventional tumor volumetric analysis, edema volumetric analysis, blood vessel density, and assessment of angiogenesis. We also calculated ADC from diffusion-weighted imaging for tumor cellularity and necrosis. And we also, because MRI covers the entire brain and is non-invasive, we were also able to detect any metastasis which could occur anywhere in the brain. So after we completed all this multi-parametric MRI analysis and scans, we also analyzed shape and texture of image segmentation, so-called radiomics analysis, so radiographic feature radiomics analysis with a specific software which is dealing with the big data, so-called large data set derived from the MRI scans. And we also, I just give you a little bit an idea, we also did some radiation. So on each of those tumor subtypes, we also did the radiation treatment, image-guided radiation treatment, mostly with 10 gray per targeted brain irradiation. And we followed up what happens after the radiation with the tumor in the brain, somewhere between 2 weeks to 2 months. So the results are, I will start with the tumor microenvironment and specific radiological features for each of the 3 tumor subtypes, medulloblastoma, high-grade glioma, pontine glioma, and ependymoma. So this is how the mouse brain looks in vivo. And this is a normal mouse brain, and you can see different compartments without any contrast, approximately 5 minutes acquisition time, cerebellum, which is obviously a very important part, especially for the pediatric brain tumors. You can see the olfactory bulb, the cortex, you can see thalamus, hypothalamus, and so on, and ventricles right here. And this is what happens when the ependymoma was injected into the 4th ventricle. It went straight to the cerebellum. And this is a small dot you see here. Can you see it well? Yeah, here. So it's around, as I mentioned before, so this was one of our smallest tumors, so it was 0.4 cubic millimeters. And you can see also that compared to the normal brain, you see the ventricles get a little bit more enlarged and involved. And this is a larger, I wouldn't say it's the largest ependymoma, but this is a larger ependymoma here. You can see it is still here, just couple days, I think couple weeks after. You can still see the same location, but it also expanded into the middle brain, and it's also metastatized a little bit into the spinal cord. And you see the bright spot here in the olfactory bulb, so it's actually also a metastasis. And you can see the edema development, those bright spots in the cortex and also around the middle brain and thalamus. So, and I just, MRI is a great criteria to see how well the tumor injection has been performed and if the tumor really homed where it's supposed to be. And so, for example, medulloblastoma, which is also location is in the cerebellum, you can see that presence here, right here. The pontine glioma is right where it's supposed to be in the pons, and this is the ependymoma once again in the cerebellum. And even like with the any fancy big data analysis, you can appreciate that the features of the entire brain is also varies from tumor type to tumor type. So after we confirmed that the orthotopic injection of the 3 different tumor types was successful, and we see it where we're supposed to see. We also looked for the other parameters and to identify what is specific for each tumor subtypes. So you can do also, as I mentioned before, for the adult human patients, you can also do so-called vascular imaging. You can see for the blood vessel density and for the angiogenesis. Actually, if you do high-resolution T2 MRI, you can see vessels, even normal vessels, quite easily. They're right here. But if you look inside of the tumor, which is usually very heterogeneous, and as you know, angiogenesis has very leaky and not quite straight capillary, right? You can do with some advanced MRI technique, you can still visualize the blood vessel density here inside of the tumor. And by doing specific analysis, you can see the increased blood flow in the tumor and also increased blood supply to the tumor. So, another physiological imaging protocol which we apply was diffusion weighted, very fast, but provides so much information. And this is, for example, how the big tumor looks like, the tumor itself, the necrotic area based to the high cell density and pressure that the cells start dying. You also can see the edema. And you can see once again, necrosis. And if you do the ADC calculation, which is a specific parameter derived from the diffusion-weighted MRI, you can characterize number-wise, so numerically, the differences in the diffusion parameters in the tumor. 0.89, very restricted diffusion because there's a lot of cancer cells. The middle numbers, 1.42 ADC number for the necrotic area because the cells start dying, so diffusion is less restricted. And finally, you can characterize edema by very high ADC, which is apparent diffusion coefficient for the edema. So you can actually create those maps as well, and you can also use those maps for the restricted, non-restricted tumor edema calculation and segmentation for the machine learning and the pattern recognition for each tumor. And you can also, based on the diffusion weighted imaging, you can also calculate the tumor vessel size, and they were actually small, much smaller than normal blood vessels, so it's approximately 0.4 to 0.7 cubic millimeters. Metastasis imaging, this is another parameter which we analyzed, quantified, and put into our machine learning pattern recognition analysis. And you can appreciate that the different tumor subtypes reveal different metastases. So, for example, in medulloblastoma, we saw a very specific appearance of those metastasis, which is actually right there in the cerebellum and the cortex, cerebellum of the tumor, in the cortex, and particularly in the hippocampus area. And it's right in the middle of the hyperintense area, which is edema. Ependymomas, interestingly, produced most of the big metastases in the olfactory bulb, and this was very consistent. So I would say the highest metastatic spread. And you usually, if you think about brain tumors, you would not expect metastasis, right? Because it's metastasis to the brain, so it's not really distant metastasis, but usually brain tumors do not metastatize, but as you can see, they still have a capacity to metastatize inside of the CNS of the system. And finally, if you look at the some of the ependymomas, but particularly for the pontine gliomas, they can also metastatize to the spine and go down and actually produce spine metastasis in the thoracic region. So this is how the spine metastasis look like. I show you a little bit different projections of the brain because there's a 3 dimensions, like in all kind of radiological science. You can do sagittal projection. If you scan the brain like this. This is so-called axial if you scan the brain like this, and this is a coronal if you scan the brain like this. And obviously some parts of the brain are visible better on coronal, some on sagittal. I always prefer to see the sagittal, especially in the mouse. I think it looks especially if you want to see the cerebellum. But for example, the spinal metastasis, they're really very nicely present and seen on the coronal slices. So after we accumulated all this data and did some analysis, we also decided to go and do so-called image segmentation. And what you do, you just delineate the area which represent, for example, peritumoral edema right here, the large tumor right here, which is in orange, and let's say necrosis right here in the middle of the tumor, which is green. And for each of those, you will have a so-called histogram. It's signal intensity based on the pixels. And our resolution was 50 microns, so we had a lot of pixel for each MR image. And you do it for the high resolution T2 imaging. You can also do it for the edema imaging with the diffusion weighted imaging. And even when we create ADC, remember those apparent diffusion coefficient for the precise tumor necrosis edema classification, you can still do this segmentation and load all tons of those histograms which represent the signal intensity of the tissue, brain, normal brain tumor or necrotic tumor or edema, into the software. And what this software does is pretty much what you probably know from the genomics or proteomic database. It just takes those quantitative data points, huge database based on the signal intensity of each MR image, and they create those clustering clustering patterns. And the machine, after the machine learning, you say, okay, this clustering pattern is, I know it is a ependymoma, and I know this is a medulloblastoma. And then when you give an unassigned image, it can actually tell you to which category this tumor belongs. And so, as I said, we had mostly ependymoma, so we had a very nice classification and classifier for ependymoma. But if you calculate sensitivity and specificity of this method, combining all image segmentation and image analysis from multi-parametric MRI, high-resolution volumetric MRI, physiological DWI MRI, and edema and angiogenesis-based DWI and FLAIR imaging, we actually get our classification for ependymoma, which sensitivity was approximately 95 and specificity 97. Especially specificity was very high for those tumor classification. And I think we have now much more animals which we have to analyze, much more images. So I think our sensitivity and specificity of this mouse brain tumor classifier would be much higher. So it is really great because it's not always easy in the real children patients to see. I mean, if you see it in for example, in a cerebellum, you don't really know if it's ependymoma or medulloblastoma. I mean, there's some characteristics which I think can be extracted from the mouse MRI, and we can try to translate it in just start doing the classifiers for the pediatric population. In addition, I'm especially interested in ependymoma because as far as I understand, and from my neuro-oncology patients, there's two different types of ependymoma. It's ependymoma A and ependymoma type B, and they actually have a little bit different molecular background, especially based on the microglial activation and tumor-associated macrophages. So if we can even do more classification of those subtypes of the brain tumors based on the MRI signature, I think it would be very useful for clinical management of those patients. And after we did all of those tumor clustering based on the big MRI fingerprints, we actually were able to more meaningfully characterize and establish a phenotype for each of the 3 tumor types. So, for example, on the high resolution T2 MRI, medulloblastoma was always hyper-intense, and the pontine glioma was especially hyper-intense. Ependymoma was very close to medulloblastoma pearl because they both are derived from the cerebellum. We saw that medulloblastoma had high level of necrosis, while glioma had a low necrosis level, and ependymoma had a moderate necrotic potential. Once again, the edema was very high in cerebellum-derived tumors like medulloblastoma and ependymoma, while the gliomas were less intrinsically infused edemic, I don't know how it is, less edema, so less swelling of the brain and water accumulation. And as I mentioned before, the metastases were also characteristic more or less for each tumor type. Very high incidence of the hippocampus metastasis from the medulloblastoma. The glioma were spreading to the spine, but not as much. And ependymoma, especially ependymoma, had this huge big lesions in the olfactory bulb, which is actually mimics exactly what we see often in children. Angiogenesis was very high in the medulloblastoma and somewhat in ependymoma, and actually glioma were not highly perfused. And diffusion-weighted imaging and ADC values, which are if you remember, is marker of cellularity. So medulloblastoma were very dense, but also very necrotic. Let me see. Here we go. And gliomas and ependymoma were less dense than medulloblastomas. So the summary on our established MAFI, multi-parametric advanced fast imaging protocol in the mouse brain tumors for PDX, where that medulloblastoma exhibit high level of necrosis and high angiogenesis. The pontine gliomas exhibit low blood vessel density and high ADC values, meaning less cell density. And I apologize for the typo. Ependymomas have high level of edema and highly metastatic spread, especially in the front brain and also in the olfactory bulb. So we used some of those animals which we used as the baseline, non-manipulated, non-treated tumors to characterize the tumor microenvironment and specific characterization of those tumors. We also use some of them for the radiation treatment. I told you that we have now the Dr. Sanakaram has an image-guided irradiator which has a CT and bioluminescence. Obviously, we cannot use bioluminescence for these PDX tumors since they don't express luciferase. But we tried to do the image-guided radiation based on the CT scans, and it was somewhat successful and somewhat not really. I think it's just because the CT has intrinsically less sensitivity to detect tumors than MRI. And obviously the irradiator does not have an MRI, but we probably will be doing the more image fusion in the future. So just to show you some of the ependymoma, and this is already published in JCI Insights. So this is, once again, this is more or less normal brain before the inoculation of the, and I apologize, this was actually medulloblastoma. It was medulloblastoma from this paper. So it was, the medulloblastoma PDX was inoculated into the fourth ventricle and went straight to the cerebellum. And you can see that, you can see even the injection site right here. Immediately, like couple days after the inoculation, and you can see the small size, and in 4 days you see the increase in size, and also due to the intracranial pressure, also the dilation of the ventricles and starting the edema process. And you can actually also see that the even overall blood supply to the brain has increased, and especially to the tumor. So on the sagittal slice and on the coronal slice. And the next animal was irradiated. And you can see that only 2 weeks after irradiation, well, first of all, radiation obviously didn't work very well on the tumor volume, unfortunately. But what you can see that the entire brain just swelled, right? I mean, it's just the shape of the brain is totally different. So if I would be calculating which I did, if I would be calculating the entire blood volume, actually it has increased almost 72%. So you can imagine how much more intracranial pressure this mouse had. But if you're looking for the tumor volume, as I said, it was a little bit of failure, and this was a CT-guided irradiation. So they tried to irradiate, delivered 10 gray jets to the cerebellum, to the tumor. So the tumor size increased from 37 to 74 cubic millimeters. But what is strikingly is that edema has increased from 18 to 43 cubic millimeter after radiation. And medulloblastoma per se is not highly inflamed, not highly edema-like tumors, so unlike ependymoma, but radiation obviously triggers this. So it triggers some kind of inflammation, even though they were SCID mice, and it triggers definitely development of edema. So by looking at the MRI for treatment response to the radiation treatment, we saw that significant increase in edema development after a single session of radiation can occur 5 days to 2 weeks after radiation. We followed up to 2 months, if we could, because actually animals got very sick and we had to sacrifice them. But there was somewhat of resolvement of the edema development, so it decreased, but in some animals, those who survived, but it definitely is peaking between 5 days and 2 weeks after radiation. So our take-home message was that we need to improve image-guided irradiation. So maybe CT in the irradiator is not good. Maybe we should use those MRI images to somehow to combine it with the CT and do MRI-guided irradiation. And we also need to assess, because the texture looks to me like a really high level of inflammation, even though I know that SCID mice cannot produce much of macrophages, but we definitely are interested in imaging radiation-induced inflammation in the future, probably using other mouse models, since we're using PDX, patient-derived xenograft. Unfortunately, we cannot use the wild types, but we're going to try to switch to the nude mice, which have some ability to produce macrophages, and to also use humanized mouse model. And for those models, we would like to use iron contrast. I think I gave it a couple times already talk. So iron contrast, iron oxide contrast, which is much less, let's say, toxic or dangerous than gadolinium. It can be used safely in the kids, and as the UK and Stanford has shown, and it can be used to image macrophages, because iron oxide contrast is usually delivered in the form of nanoparticles, and macrophages love both, they're attracted both to nanoparticles as well as iron. And while cancer cells don't care about nanoparticles or iron. So if iron is delivered to the tumor and there are macrophages, either intrinsically existing tumor-associated macrophages or radiation-associated macrophages, RAM macrophages as I call them, then the iron contrast will be taken by macrophages, and the T2 MRI will change its intensity, so you definitely can quantify how much iron is in the tissue, which will give you an idea how much of the inflammatory macrophages is in the tumor. Which brings me to the, and it's very, very brief overview what we've done so far, not in the brain, because just for the developing of the contrast protocol is easier to do with the flank xenografts, but we use SCID mice with the flank xenografts, and we did 10 gray irradiation. And you can see that before injection of the contrast and post-contrast, there's almost no changes in the tumor. And the reason for this is because even after radiation, SCID mice cannot produce macrophages, and the cancer cells don't take iron, so there's no contrast accumulation in the SCID mice with the GBM tumors. However, if you take nude mice, which already have some ability to produce macrophages, and you first of all, you can see that the purple is where the iron, where the low level of the inflammation is, but you can see, about low level of the cancer cells and more macrophages is, you can see that after the radiation in nude mice, you can have significant decrease in T2 relaxation time, meaning increase in the purple color. So this is regions where the iron was taken up by macrophages. So the nude mice definitely have an ability to produce some macrophages after radiation. And we also did, most recently, we did a wild type inducing the murine glioblastoma cell lines into the flank. And you can see that without radiation, after injection of iron contrast, we see a little bit of the iron accumulation, intrinsic tumor-associated macrophages, but after irradiating those tumors with the 10 gray, the pre and post-contrast show significant accumulation of iron in the tumor-associated. It's actually not tumor, it's probably radiation-associated macrophages, because as you know, there's a different type of macrophages, M1 and M2. So we are working now on developing a humanized PDX model where the neuro-oncology team can inject those pediatric PDX specimens in the brain, but we will have a mouse which is able to produce human macrophages, and so we'll see how the response, inflammatory response to radiation will go along with the development of edema after the radiation treatment. And obviously it will be very interesting to see if some checkpoint inhibitors or other kind of immunotherapy can be synergistically applied together with the radiation in treating the pediatric brain tumors. So, and I think actually I'm pretty much done with this. Once again, I'm just really fortunate to have a wonderful team working with me. And so my own team, you probably heard last week, Jenna was presenting about the animal imaging. Jenna is our veterinarian certified expert, so she's really doing a wonder with the animals during the imaging session. Also, Alina just started with us. You probably know Kendra she was with us for almost 12 years. She's doing mostly human imaging right now. Melissa Cardboard worked very closely with us on all aspects of the animal imaging and animal handling, and also my student Heather, Paul, Aaron, Sam, and Marina will be joining us in the summer with a scholarship to actually continue on those MAFI and machine learning and image segmentation analysis on the pediatric brain tumors. And my special thanks go to Dr. Angela Pierce. She is phenomenal on making those orthotopic brain tumors. I mean, she's as precise as any of neurosurgeons. It's really probably even better because the mouse is smaller. So it's really impressive. And Andrea Griesinger with Nick Foreman team, Bethany, Aaron Knox, and Nathan Dahl, who's our neuro-oncology resident. So they're all doing a great job with the mouse models, and also our radiation oncology team, irradiator team. So, and most of all, I just thank our four-leg patients here for being so patient with us, and don't holding grudge. So if you have any questions, I will be happy to answer. Thank you.
This is a very naive question. The biggest difference between the three types of tumors we can see to be between gliomas and the other two. And they were injected in the same site. I realize these pediatric tumors are very precious. Have you ever tried injecting any of the gliomas into the ventricle and see more mimics the other properties?
So, unfortunately, I won't be able to answer this question. I don't think Angela is here. There are some neuro-oncology folks are here. Do you want to answer the question? Okay, so the question is, it seems like glioma is really very different from ependymoma and medulloblastoma based on our data sets, our classification.
So the question then was, is some of these measured properties more a function of where they were injected? Because two types that were similar injected in the ventricle, whereas they all were
Well, but you know, it's almost like if. But this is why we did it, because they belong there, right? So it's almost like you will inject prostate cancer into the mammary gland and ask the question, are they going to be the same with the breast cancer, right? You don't want to do this. You want to inject it in the prostate. You see what I mean? So I think maybe intrinsically there's a explanation why they are so different, because the home intake is in the different part of the brain, and it's why you're injecting into the different compartments.
So I can answer that a little bit.
Oh, Rajiv, thank you.
The issues, for example, if you take a medulloblastoma and inject it in the cerebellum or inject it in the cortex, they will grow in both locations, at least the more established xenograft model. But if you then take them out and, for example, the gene expression or methylation out, whatever, they're actually very different. Whatever is local to that location actually alters the biology of the tumor. So even though the tumor will grow in a different location, the biology is different. If you take a medulloblastoma in the cerebellum that metastasizes to the cortex and down the spinal cord, genomically those are also different from the parental tumors expression out. So there are differences that are induced by the environment of wherever the tumor happens.
So you talked about that classification you had and the fact that you could tell with 95% accuracy or sensitivity or whatever, 97%. I'm trying to remember which one that was. That was your, not medulloblastoma, but but not your DIPG, that's the one I'm wondering about. Getting the, I can't say that one, epidymoma or whatever.
Ependymoma. Actually, this is ependymoma. I like the word.
So the DIPG looked to me like the pattern was sort of across all of them and didn't look to be as, you know what I mean? Like you have a number of DIPGs that are falling overlapping the other lymphomas.
Right.
Some that are even out towards medulloblastoma. So how accurate can you be to predicting those? Because it seems to be overlapping. There's a lot of mistakes in it.
Right. But I think if there's very if you do all kind of those analysis and like PLSDA analysis and machine learning analysis, there probably will not be the absolutely separation of the groups. It's always together, right? So I think if you have this point right here and you will be wondering if it's ependymoma or it is a DIPG, I think what you probably should be doing afterwards, you can, and it's probably what brings your specificity and sensitivity for DIPG down to like, I think it was 82%. You probably just will go back and just check more quantitative data set and see if you can find more characteristics, specific characteristic that, right. But there will always be mixed classification, and they're always all even will be all outliners. I mean, there are some outliners in each study using those machine learning and big data.
Well, that graph on the next slide is based on food, or was it based on all?
Based on all features. Mm-hmm. Yeah, so obviously, right. And so you look here now for function 1 and function 2, right? It's like the discriminative analysis, but you can also have a 3, 4, and 5. So you can make it 3-dimensional, 4-dimensional, absolutely. And it's yeah, and you probably can answer the question which Heide was asking. Not being a really bioinformatic person, I would rather go to my quantitative data set and do it like hard way. Yes. Thank you, Phil. Right. So it's why I was actually saying because we scanned so many animals from Rajiv's group, I tried really, I mean, for this specific one, I tried to do, well, actually, they were all non-treated, and I tried not to have a very large tumor size, variation. So it was like from very small, I mean, 0.4 is what we detected, but it's usually like around 30, 35 cubic millimeter to maximum 70 cubic millimeters. And I think your tumors can grow even bigger, right? But then, so this is something to look further, because I agree with you. Probably the bigger they get, the more metastatic spread they will have. So I think it's important to have as a function of the tumor size, I guess, but we haven't done this analysis yet. So I was trying to make them the mean tumor volume relatively the same.
Does the software co-register that ROI so that it follows it when you put the mice back in every time?
Right. Yeah.
And we do have a little bit of longitudinal data from that academic program we had treated and when we have over time treated or untreated to show changes in the body. So we have data for that. The metastatic part is really important and interesting. We can see if there was a risk tag, we can see that happen, but it's not very clear. And so if we can get the MRI graph to really track the metastasis, that will make it maybe different. That's really the limitation that we have with these models is that they, because they're in the cranium, there is that limit to intracranial pressure where the animals die. So we only have a limited, like for example, the medulloblastomas untreated all die about 24 days post inoculation. So we have to figure out how to manage that, for example. So there's some limitations to the models themselves that we have to think about.
Right, and we also, I mean, from Nick group, we also have a lot of longitudinal studies and scans from the ependymomas. I mean, they go like really from 0.4 to like the mouse head is just even does look normal anymore. Okay, thank you.